I'm Cody Reightley, and I hate presentations.
I'm Abeyta, and I occasionally ride the Abeyta bus.
And this is CRISPR than an apple.
All right, so what is CRISPR first? CRISPR is clustered regularly interspaced short palindromic repeats, which is a lot. But basically, it's repeats with these spacers in the middle that are basically the viral DNA that bacteria has picked in. And so it's this CRISPR acts as the immune system for bacteria.
Okay, so let's say bacteria is just going around, and then a bacteriophage comes in and it infects it. Well, the CRISPR will take the RNA that is injected into it, and it will actually take it and incorporate it into its own DNA. And by doing that, it actually creates a piece of DNA that will code for an enzyme that will then break down the RNA that would have been put into the bacteria so that it won't be infected anymore.
This is basically showing how the enzyme does it. So it goes on there, on the enzyme, the RNA, the viral RNA, and it breaks it apart, and then donor DNA comes in and fixes it, but since it's messed up already, it doesn't function, so it can't make the virus, the virus can't do anything to the bacteria.
So you get your bacteriophage there, it's infecting it, and then this is actually the enzyme that would incorporate it into its DNA, which then creates the spacers right there.
So they're using this technology to try and get rid of many types of viruses like hepatitis and HIV. And how they're doing that is they're having the CRISPR try and locate or stop the RNA from being sequenced or break it down so that the HIV can't use the RNA in the cell. But right now they can't. They've only been able to do it in single-celled organisms. They're trying to figure out how to do it in large scale.
The research findings we are now reporting could be a game changer in the fight against HIV and AIDS. Our goal is to cure AIDS, and the time is now. More than 33 million people worldwide are infected with HIV-1, including more than 1 million people in the United States, and every year another 50,000 Americans are infected with HIV. Current therapies have transformed AIDS into a chronic illness, but the root of the problem, the virus, has not been eliminated, only suppressed. Our goal is based on a simple concept. If you want to cure a viral-based illness, eliminate the virus. My colleagues and I developed molecular tools that permitted us to successfully eradicate HIV-1 from cultured human cells. We have the technology, and our findings are proof of concept that we are moving in the right direction. Our approach, which is still years removed from the clinical setting, would not only eliminate the virus permanently, but also create a protection against a new infection or reinfection. The molecular tools developed consist of targeting strand of RNA called guide RNA and the DNA snipping enzyme. When deployed simultaneously, the guide RNA hunts down the viral genome and the snipping enzyme takes over, eradicating completely the HIV-1. The newly virus-free cell then repairs itself. By discovering a way to permanently rid human cells of HIV-1 in a laboratory setting, we have set the stage for additional preclinical work that we believe will lead to a permanent cure for HIV and AIDS. The clinical application of our research may also serve as a therapeutic protection against the HIV-1.
We believe this research has set the stage for the development of a potential cure for patients with AIDS, as well as for patients with other viral-based diseases. However, I think this also shows the importance of federal investment into research in academic medical centers, because without that investment, research of this type would be impossible.